Hello everyone, hope you all are doing good. In today's topic, we are going to discuss about antimicrobial action of MYRISTICA mother tincture as well as potencies against the isolates of suppurative infections, a in vitro study. In this particular work was done by Dr. Vivek Sakthidharan as a postgraduate work under the guidance of Dr. Pravas Kumar. The work was done in Mangalore Homeopathic Medical College, Mangalore. Through the study, they have aimed to find out whether the MYRISTICA mother tincture and various potencies have an action against the pathogenic microorganisms or not. That is one aim. Another work is they wanted to compare between the mother tincture and various potencies. So these are the ideas behind this particular project. For this work, they have collected microbes from deep and superficial infections, pathogenic infections, and they have cultured, processed it and identified what are the microorganisms available under laboratory of microbiological laboratory of Muller Medical College. Further, they have worked on antimicrobial study. For bacteria, they have selected Muller-Hilton agar, and for fungus, they have used Seborrheic Dexter agar. After culturing in the respective agars, what they have done is they have adjusted the solution to 0.5 McFarlane opacity tube, and then they were subjected to further antimicrobial analysis under 3 methods. The 3 methods used are disk diffusion method, punch well method and agar dilution method. So using these 3 methods, they have identified the antimicrobial action. Standard protocols were followed for all the 3 methods. Simultaneously, they have also collected ATCC cultures of Staphylococcus, E. coli and Pseudomonas and also have performed these analyses. The observations noted were in the disk diffusion method as well as in punch well method, they did not find any inhibition. So there was no antimicrobial actions noticed in these both methods. But in case of agar dilution method, they have noticed a minor differences or inhibitions, but not in the potencies. Only for the alcohol and mother tincture, this particular identification was made, but not for the potencies. The microbes isolated from 100 pathogenic infections are Acinetobacter, Candida albicans, Citrobacter, E. coli, Enterococcus, Klebsiella, Pseudomonas, Proteus, Staphylococcus and Streptococcus. The maximum occurred or recurred pathogens were Staphylococcus, Klebsiella and E. coli as well as Pseudomonas. The results obtained in the antimicrobial study was in the alcohol group. For Pseudomonas, out of 23 samples collected, 3 had moderate inhibition and 4 had mild inhibition. In mother tincture group, 3 had moderate inhibition and 1 was having a high inhibition and 6 had mild inhibition. For Klebsiella, in alcohol, 1 had mild inhibition out of 15 cases. The same way, in mother tincture also, 4 had mild inhibition out of 15 cases. In Enterococcus, mild inhibition was noticed in 1 out of 3 patients and as well as in the mother tincture group 1 out of 3. In the Staphylococcus aureus, mild inhibition was found in 1 patient out of 33 samples collected. In mother tincture, mild inhibition in 2 out of 33 collected. In E. coli, Citrobacter and Acinetobacters, there were no inhibitions noticed in alcohol group. In mother tincture group, E. coli showed 2 inhibition out of 11 patients as well as 1 inhibition in Citrobacter out of 4 and in Acinetobacter 1 inhibition out of 3. So overall, when we club the results, we get to see that in the gram-positive bacteria, 11.67 were resistant to alcohol and 0.67 were mildly inhibited by alcohol. In gram-positive, for mother tincture, 11.33 were resistant and 1 showed mild inhibition. In gram-negative bacteria, 8.33 were resistant and 0.83 were showing mild inhibition and 0.15 were moderately inhibited by alcohol. But in case of mother tincture, 6.67 were totally resistant, 2.33 were showing mild inhibition, 0.5 showed moderate inhibition as well as 0.17 showed high inhibition. In fungus, there was no inhibitions noticed. So overall, by seeing the result, what we come to an understanding is there is mild inhibition both by alcohol and mother tincture. Mother tincture having a slightly superior inhibition action compared to alcohol, but not statistically significant. So this is the understanding we come across by this study. Hope it was useful and informative for you. Meet you all again with another interesting research. Take care.